When it comes to metabolism, things are messy, but they're logical. And so rather than trying to memorize pathways and that sort of thing, let's focus on the logic behind these pathways and the logic of metabolism, the logic of how we have all of these interconnecting pathways, all of this crazy stuff going on. But really at its heart, we can focus on things like the chemical logic. Why are things happening in this order? The thermodynamic logic. How can we use favorable reactions to drive unfavorable reactions? How can we skew the concentrations of things so that we're always going to have a drive to go one direction or the other, or so that we can change direction so that we can use the same enzymes to go in multiple directions? How can we do that? Basically, we can do that in a variety of different ways. We can regulate the concentrations by doing things like compartmentalization, by using things happening in the mitochondria or things happening in the cytoplasm, things happening in whole different organs and cell types, where we can regulate the concentrations of these things differently to control the direction of a pathway. Within the pathway, concentrations can only get you so far, especially if you have a reaction with a really big delta G naught. And so for those, what you want to do is basically regulate the enzymes. We'll see instances where we have, say, the liver has enzymes that other organs don't have. This is why we can do gluconeogenesis. We can make glucose in our liver, but not in other tissues, because the liver has the enzymes that we need in order to do this, but those other tissues don't have that. We can also basically just kind of regulate the availability of those enzymes to actually be where they need to be. So in addition to having them in different cell types, we can have them in the cell type kind of be sequestered, maybe in internal like vesicles, and then only put them on the surface when we're right, when we get the signal. So hormones are going to be a really big thing too. And then when we have the enzymes, we can regulate their activity. So we can regulate enzymes on a variety of time scales. So one, like we talked about before, like that would be slow, would be kind of like transcriptional control. Like, do we actually have the enzyme present? A bit faster is if we've got it present, we've got it made, but it's kind of being sequestered. If we want to really quickly change the activity of an enzyme, we can do this in a couple of ways. One is by covalent regulation. This is typically done by phosphorylation, where we can say have a phosphorylation cascade set off by a signaling pathway that's going to then regulate the activity of these enzymes and therefore regulate the pathways. We'll also see how we can have allosteric regulation. There, instead of having a covalent modification to our protein, instead of having something like a phosphorylation, we're basically just having our enzyme bind to a small molecule. And when our enzyme binds to the small molecule, we can get an allosteric change. The protein is going to basically undergo a shapeshift that's going to make it either active or inactive. And all of this can regulate what pathways are happening when, which direction different intermediates are being siphoned off into. So remember, we basically call everything an intermediate because even if it looks like it's at the start of a of a pathway, or is at the end of a pathway, really we don't have pathways. We have this giant complex subway system where things are happening. Things are going siphoned off into one direction or the other. But if we're looking at one of these giant big maps, it can get really, really overwhelming. And so we like to have some sort of like simplified logic. We can think about things in terms of pathways rather than thinking about them in terms of this giant web. But we need to always remember that we are in the context of this giant web. So although we're going to talk about like cellular respiration as being, okay, we take glucose, we take our blood sugar, and we're going to break it down. We're going to go through glycolysis, turn it into pyruvate. We're going to go through the citric acid cycle, that Krebs cycle. We're going to basically break it down even further, get a bunch of electrons out of it, pass those through NADH and FADH2 into our mitochondria, where we can then cash in those electrons through oxidative phosphorylation to get ATP. And it seems like it's this nice, pretty pathway where things go in one direction. But even if we think about it as a pathway, that pathway can go in both in two directions. Not exactly the same, but basically we can think about glycolysis, the breakdown of sugar to get to this pyruvate step. We can think of this pathway as we can kind of reverse a lot of the steps. In fact, we can reverse like 7 of the 10 steps. And when we do this, we can make glucose through gluconeogenesis. But there's also going to be steps that we can't easily reverse, and these are going to be sites of key enzymatic control. And all of this, though, is happening in the context of all those other pathways. So not only do we have glucose and glycolysis and gluconeogenesis, but we also we'll consider things like, well, what about the pentose phosphate pathway? It's an alternative fate for glucose. How do we decide what goes there? And later in the course, we'll look at things like, what if we want to go make or break down fats or things like this? So remember that all of this is interconnected, and let's try to focus on the logic. Let's try to use this glucose versus glycolysis as kind of, although we're talking about it kind of like it's an either or, this is just kind of like to give us a little example so that we don't get too overwhelmed by the giant picture. But remember that we're in the context of the giant picture, and things are often going to be more complicated than we're going to make them out to be. That being said, let's try to simplify things down and think about this glycolysis as a pathway and gluconeogenesis as a pathway that's going kind of the opposite direction, but it's going to take some detours. So just like when we were talking about kind of thermodynamics and metabolic logic, the logic of kind of pathways and things, we talked about how there would be steps that would be close to equilibrium, and then there would be steps that would be far from equilibrium. We talked also about how there was this G naught, or the delta G naught. Basically, we said we wanted a negative delta G in order for a reaction to be favorable in the forward direction. Now, a delta G naught is basically saying it's kind of inherent. A negative delta G naught would say it was inherently favorable. But then we talked about how the actual favorability was determined not only by that delta G naught, but also by the concentrations, the amount of your products and the amount of your reactants. We talked about how even if you had a really inherently favorable reaction that had a large negative delta G, that if you had enough of your products, you would actually drive things backwards. But the bigger that delta G is, the more skewed your concentrations have to be in order to do that. And even if you had your concentrations really skewed, they wouldn't be that way for long because you'd be driven towards equilibrium. And as you drive towards equilibrium, you lose that drive. So that's why we keep a steady state rather than getting things to equilibrium. We maintain fairly constant levels of things by replenishing what we're taking out. And then we can take out things in order to drive pathways forward as well. So the actual G, the actual free energy change, is going to be different than the G naught, sometimes dramatically so. That being said, if we look at the G inside of our cells, we can see in the glycolysis for the different reactions of the glycolysis. So basically, this is going to be saying in erythrocytes, so in red blood cells, they went and they calculated the actual delta G for each of the steps of glycolysis. And we will review in a second the steps of glycolysis and the logic behind it that we got practice with our activities in class. But basically, we have these 3 steps. This first step where we have glucose to glucose 6-phosphate. This third step where we have fructose 6-phosphate to fructose 1,6-bisphosphate. And we have this phosphoenolpyruvate to pyruvate and ATP. And these 3 steps are going to have these large negative delta Gs, which means that if we were to try to go in the reverse for these reactions, we would need to have concentrations so skewed that basically it wouldn't be possible in the context of our cells. We call these metabolically irreversible. Basically, we can't reverse them just by changing little bits of concentration wise. We need to actually control the enzymes that do this. These 3 steps, basically, so we have this hexokinase step, we have this phosphofructokinase step, And we have this pyruvate kinase step. These are going to be the 3 steps that have that really big negative delta G. So those are going to be the steps that if we want to do gluconeogenesis, if we want to make glucose, those are the steps that we're going to have to go around. Those are the steps that we're going to have to bypass using different enzymes. We're going to see how we can use different enzymes and regulate them reciprocally. So for example, we have the first reaction of glycolysis is carried out by hexokinase. And if in gluconeogenesis, the kind of opposite reaction is going to be carried out by glucose-6-phosphatase. And these can be regulated reciprocally so that when we turn off hexokinase, we turn on glucose-6-phosphatase and things like this. Similarly to phosphofructokinase 1, which is going to basically take fructose 6-phosphate and phosphorylate it again to give us fructose 1,6-bisphosphate. In the opposite direction, instead of doing things, just taking this enzyme and going reverse, we're going to actually just use a different enzyme, fructose 1,6-bisphosphatase 1, or FBPase. Now, it turns out that this enzyme is actually going to be super duper highly regulated because this is going to be kind of like our first committed step of glycolysis, because once after this glucose 6-phosphate step, after we phosphorylate glucose and kind of like trap it in the cells, well, then it can be used for various things, including like being used for the pentose phosphate pathway, being linked up to form glycogen and things like this. But once we go to this step, this is kind of going to be, we're committed to glycolysis. And so we're going to want to heavily regulate this step. We'll see how we can regulate these enzymes in various ways. Sometimes we're going to regulate them by using like products that are later down the line to kind of like inhibit yourself with the thing that you make so that you don't build up too much. Like you're like, okay, we've made enough now. Our supply is exceeding our demands. We can also respond to our demands. So we can have allosteric regulation by things like ATP and ADP and AMP so that we can respond to energy levels. So we're going to see how all of this is going to come into play. And then finally, the last step that we're going to need to overcome is going to be this step from phosphoenolpyruvate to pyruvate in the glycolysis direction, and that's carried out by pyruvate kinase. To go in the reverse, we actually have to use 2 enzymes. So we're going to have one extra enzyme in our gluconeogenesis step because we're going to have to use pyruvate carboxylase to go from pyruvate to oxaloacetate, and then PEP carboxykinase in order to go from oxaloacetate to phosphoenolpyruvate. So we have to kind of take 2 steps because that reaction was super duper favorable. And so we'll talk more about this when we get there. Let's start by thinking about the logic of the glycolysis pathway as a pathway, though, and kind of review that chemical logic. So why are things happening in this manner? Like, why do we move things, put a phosphate here? Why do we shift things around? It's not just random. It has to do with what makes it then chemically favorable to do the next step. So in glycolysis overall, we can kind of think about it as being split into 2 phases. An investment phase where we have to put into ATP, and a payoff phase where we're basically going to get for ATP as well as to NADH, which remember can then go into the mitochondria, go through that electron transport chain and give us ATP. And ultimately, that's where the majority of our energy is going to be coming from, is actually from that oxidative phosphorylation, assuming we have oxygen around. But first, we have to put in that investment. And so that conversion of glucose to pyruvate overall is going to be exergonic. So it's going to let off energy. And so basically for any pathway to work, it has to be exergonic. But that doesn't mean that every step is going to be super duper favorable. Instead, basically, the formation of ATP from ADP in organic phosphate is endergonic. But yet we're going to do that in our pathway. We're going to make ATP, and this is not going to be favorable, but because we couple it to favorable things, we make it favorable. And so this is what we talked about before, this idea of coupling of reactions. So you can see that here we kind of like split things in two. So when we generate this ATP, we're actually generating two ATPs. So one ATP here and one ATP here, and then we're generating 4 ATPs overall because we split that in two. So we do each of this 2 times, but then we use 2 ATP. And so that's why we end up with a net gain of 2 ATP and 2 NADH. By coupling all of this, basically what we're able to end up with is basically our standard free energy change for the overall reaction is going to be minus 85 kilojoules per mole. So the pathway makes sense. We're able to use this super favorable reaction, to power a non-favorable reaction and overall still have a favorable pathway. So let's talk a little bit more about the pathway and the enzymes involved in their regulation. The first step is going to be taking glucose and phosphorylating it. This is just a simple kinase reaction, except that this kinase is going to be regulated, as we'll talk about. But this is hexokinase. It's also in our liver, we have a form that's called glucokinase, and we'll talk more about that later. But basically what this is doing is it's trapping the glucose inside of our cells. Remember how our membrane is going to be hydrophobic, and so it's not going to just let glucose, it's not just going to want to let a phosphorylated glucose through. And so what we're going to have to do is we're going to phosphorylate it and trap it inside of our cells. Now, we wouldn't want this reaction to be easily reversible because then we would kind of just be letting that glucose flow back out of the cells after we trapped it. But we want our liver to be able to release the glucose that it makes to give it to other cells, because remember, gluconeogenesis is happening in our liver, but other cells are going to need that glucose. And so basically we have this, this other enzyme, glucose 6-phosphatase, that's basically going to hydrolyze that phosphate. It's going to basically use water to break it off. Instead of trying to put a phosphate group back onto ADP, from the glucose 6-phosphate, it's just going to cut that phosphate off. And so this is glucose 6-phosphatase is the enzyme that's going to do this. And it's in our liver, in like a special place in our liver, so that basically it's not going to let the glucose out if you don't want to. So that's going to be highly regulated. And so this is kind of like our first regulated step, although we'll also talk about how just getting the glucose into cells can also be regulated. But this is carried out by hexokinase, and it's going to put this glucose on the 6th position. Sorry, it's going to put this phosphate group on the 6th position. And note that when the kinase is transferring phosphate, it's really transferring a phosphoryl group. So this oxygen here is the one that came from the glucose. And then this phosphoryl group, so the phosphorus and 3 oxygen groups, not with 4 oxygen groups, that's what's actually going to be transferred. So this is not an oxidation, but this is a phosphorylation, and it's carried out by hexokinase. And we can see that it has a very large negative delta G. So this would not be easily to reverse, which is why we're going to use this different enzyme in gluconeogenesis. Okay, what about the next step? Well, here we just have an isomerization reaction. We're not changing the number or the type of atoms. We're just shifting things around. So we're basically moving the position of this carbonyl, and this is going to free up this end-hydroxyl group so that we can phosphorylate it. And so the enzyme that's going to do this is phosphohexosisomerase, sometimes called phosphoglucosisomerase. So you'll see it listed, you'll see it called both of these things. You can see that it's got a pretty neutral delta G, so this is going to be easily reversible depending on things like the concentrations. And we'll see later how this F6P can also be changed into further phosphorylated to give us F26BP, which is going to be an allosteric modulator. But for now, just focus on the fact that we're basically taking this glucose 6-phosphate and making fructose 6-phosphate, which just involves shifting of this carbonyl group. You might be wondering, why the heck are we doing that? Well, it turns out that doing that, we basically free up this oxygen so that we can phosphorylate it and then have this molecule we can have this molecule be cleaved in half and have a phosphate group on each end. And by moving the position of the carbonyl, well, then it's going to set it up so that when we have a reaction later, it's going to basically cut the molecule in half rather than cutting it in uneven parts. Our next step is going to be phosphorylating that hydroxyl group that we just freed up. And so this is going to be that super duper regulated step. The step of phosphofructokinase, or PFK1. And so this is going to be highly regulated because it's going to be that kind of committed step of glycolysis. And so we'll talk much more about that later. But basically here, you're sticking a phosphate group on the other end. You're sticking a phosphate group on this one position. And you're taking that phosphate group from ATP. And so this is being carried out by a kinase, PFK1. It has a very large negative delta G, minus 22.2 kilojoules per mole in erythrocytes. And so this is why we're going to need to go take a different path if we want to reverse it in the case of gluconeogenesis. And we'll talk about that in a minute. But continuing on our story of glycolysis, the next thing that we're going to do is we're basically going to split that molecule in two. So in this step, basically, the purpose of phosphorylating the second end was so that we could split it in two and have two parts that had the phosphate groups, and that these parts were basically interchangeable. And so we'll see in a minute how they can interchange, but first we need to split it in two, and that's going to be this enzyme called fructose bisphosphate aldolase, most commonly just called aldolase. What it's going to do is it's going to take that fructose 1,6-bisphosphate and it's going to cut it in two. This is going to give you two molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is going to have a pretty neutral delta G, which means that this is going to be we can go in either direction. We can use this step in both glycolysis and gluconeogenesis. Once we get these two products, well, now what actually happens is that they're going to turn out being basically the same thing, because we're going to have this enzyme, triose phosphate isomerase. Basically, you're going to go from DHAP to glyceraldehyde 3-phosphate. This reaction, although it's kind of like really easily reversible, what's going to happen is that then you're going to use these favorable steps that are going to pull glyceraldehyde 3-phosphate through the pathway. And so you start out with having both these dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. You start with equal amounts of these, but then as you pull glyceraldehyde 3-phosphate away, well, now you're basically going to drive the concentration of that down, which is going to pull the dihydroxyacetone phosphate towards that good old Le Chatelier's, and that's going to make it so that we can pull this reaction forward and just work with this glyceraldehyde 3-phosphate. Note that once you split it in two, you can't really tell what came from what. And so you have these two equal parts, but now everything in this kind of like payoff phase is going to be doubled. And so everything that happens down here, we're going to double it. So that's why we get like 2 NADH instead of just 1, because all of this is happening twice because we split the 6-carbon molecule into 2 identical 3-carbon ones. Well, not identical at first, but then identical. Now what we want to do is we're going to enter that payoff phase. We're finally going to start making energy money, but first we get an IOU in the form of NADH. And we pick up a free phosphate. Okay, so when we see NADH, what we should be thinking is something's going to be getting oxidized because we're reducing NAD plus in order to form NADH. And in this case, we're also going to be phosphorylating. So we have oxidation and phosphorylation. We have an oxidative phosphorylation. And in fact, this is so you can see the reason why I was saying before that a kinase was going to add a phosphoryl group was because here we're actually adding a whole phosphate group. So we're adding 4 oxygens. So we are oxidating this, and we're going to see that that's an oxidative phosphorylation. Now, typically when we think of oxidative phosphorylation, we're thinking about that thing that happens in the mitochondria where we're passing off all those electrons. And that is oxidative phosphorylation, but there you have the oxidation and the phosphorylation separated, whereas here it's happening at the same time. This is happening into this molecule glyceraldehyde 3-phosphate. We're making 1,3-bisphosphoglycerate, and we're doing this using this enzyme glyceraldehyde 3-phosphate dehydrogenase. So as the enzyme name suggests, this is going to be an oxidoreductase enzyme. And this is consistent with the idea that we're adding a phosphate group and we are oxidizing in the process. So this is going to have a neutral delta G. So again, we can use this in either of these directions. Next up, what we're going to do is basically, we're going to take off the phosphate that we just added. Now you might be like, why the heck did we do that? We just took this phosphate group that was freely floating around and we added it onto glyceraldehyde 3-phosphate, and now we're taking it back. Well, we're not just cutting it off. Instead, we're going to be adding it onto ADP to give ATP. So why could we do this? Well, it turns out that we're basically adding we're making a more energetically, like high energy phosphate bond than we would have with like ATP. And so we're able to add this phosphate group onto ADP in order to get our 3-phosphoglycerate. Why we can do this is because basically what we have is we have this really high energy phosphate bond where we have this acyl phosphate. So this is an acyl group where you have a carbonyl next to basically just like a carbonyl attached to something. And here that something it's attached to is a phosphate. So this is an acyl phosphate group, which is going to be high energy. And so breaking it off is going to give us a lot of oomph, and it's going to be favorable. So we're going to be able to then take that phosphate group off of the 1 position, leaving us with 3-phosphoglycerate. So we went from 1,3-phosphoglycerate to 3-phosphoglycerate. And of course, our ATP that we stuck the phosphate group off of 1,3-bisphosphoglycerate onto ADP to give us our ATP. And remember, that's happening twice because we have this whole second part happening twice. Now what we're going to do is we still have that 1 phosphate group, so we still have that ability to make 1 more ATP, but right now it's not in a good place to do so. It's not a high enough energy bond in order to make ATP. But we can set things up to be powerful enough by using phosphoglycerate mutase. Basically what we're going to do is we're going to shift the position of the phosphate over to the middle. And so you might think, okay, well, what's the big deal about sticking it in the middle? The big deal is basically what we're going to end up with is we're going to be setting ourselves up to form a very, very favorable interaction. So this one's not that exciting. We have this really, really neutral delta G. But basically, we're able to take this, make 3-phosphoglycerate, move the phosphate group to the 2 position to give us 2-phosphoglycerate. And now we're going to set ourselves up for the big payoff. So basically, next step we have is basically now that we've moved things like this enzyme enolase, what it can do is it can do an elimination reaction to remove water. When you remove water, well, now what you formed is this enol. So hopefully you remember back to your OChem where you had an enol was going to be like an alkene, so a double bond between 2 carbons next to an alcohol group. And so enol, you can get this kind of like tautomerization where you switch between an enol form and a keto form. So like a ketone, if you think about like moving these electrons over to here, and then just pushing things up and moving over a proton, basically you can switch between these 2 forms. This keto form is going to be much more favored than the enol form. So if we just take like an enol and we let it go, it would like really want to go to the keto form. But what we're able to do, what enolase is able to do is it's basically going to trap it in this by that phosphate group is going to allow it to kind of trap in this enol form. And when you are trapped in this enol form, basically what's going to then happen is it's going to want to go back to the keto form. And so it's going to really, really, really want to go back to the keto form, so much so that we're going to get this negative 16.7 kilojoules per mole of energy if we let it do so. And so we're going to let it release that phosphate group, transfer it to ADP to give us ATP. This is going to then give us this energy that's kind of like the Big Bang at the end of our pathway. And this is going to give us another ATP, which again, we're going to have all of this happen twice. So that's why we get 2 ATP here, 2 ATP here, but we use 2 ATP here. And so basically then we're going to get a net of 2 ATP and 2 NADH. And we get 2 pyruvate, which can go in the citric acid cycle and get further oxidized. But that's only going to happen if there's oxygen around, because that's going to require the oxygen to be our terminal acceptor. But if oxygen isn't around, then we're going to run into trouble, because one of the things about the citric acid cycle was that it was also going to regenerate our NAD+. And so if we don't have NAD+, well, then this whole pathway isn't going to work, because we needed that NAD+ here in order to do this step. And so if we don't have NAD+, well, now we're in trouble. And so we need alternative ways to generate NAD+ if we don't have oxygen around. And this is what we get with like fermentation. And so that's going to regenerate NAD+ without oxygen so that glycolysis can continue anaerobically. So in some yeast and plants and microbes, they're basically going to be able to take things to ethanol. But we're going to take those pyruvates and change it to lactates. So this is done by lactate dehydrogenase, and that's an alternative way to generate NAD+. Another kind of alternative way to generate NAD+ is to basically make glucose, because if we do the reverse pathway, well, then we're going to basically generate 2 NAD+. But this typically isn't like the main reason why we would do gluconeogenesis, but we do get NAD+ out of it. So let's talk about when we would want to do gluconeogenesis. Basically, you would want to do gluconeogenesis when you want glucose. So when you've got plenty of energy, but not much glucose. And when would you want to do glycolysis? When you've got lots of glucose, but not enough energy. And so later we'll see how these are kind of reciprocally regulated by things like ATP levels, ADP and AMP levels, NAD+ and NADH levels. And so we'll see that the enzymes responsible for these kind of bypasses, those steps that we can't easily reverse, are going to be reciprocally regulated largely. So let's introduce these enzymes, these bypass steps, and then we'll talk about their regulation and more. As we mentioned at the beginning, you basically have these 3 steps with these large negative delta G's that we need to get back the other direction. And so when we're doing this in gluconeogenesis, we're basically going to have to introduce different enzymes. We're going to have we're going to have 4 enzymes because we needed 2 enzymes to basically reverse this last step. So this last step, remember, that was in glycolysis. We are going from phosphoenolpyruvate to pyruvate, and that was done by our pyruvate kinase. It was going to be really, really favorable because we have that whole enol keto tautomerization thingamabob. And so we were able to put a phosphate group onto ADP to give us ATP. And that was carried out by pyruvate kinase. To go in the other direction, basically, we're going to need 2 enzymes in a kind of convoluted way. Basically, we're going to take pyruvate, we're going to change it to oxaloacetate through pyruvate carboxylase, and then we're basically going to take that oxaloacetate and use PEP carboxykinase in order to phosphorylate it to give us phosphoenolpyruvate. So we end up having to actually pay for each pyruvate, we're going to have to pay an ATP and a GTP. You need to put more energy in for gluconeogenesis than we get from glycolysis. But basically, we have these 2 steps, and to make things even more complicated, they're happening in kind of different areas. So basically, we're going to carry out part of this in the mitochondria and part of it in the cytoplasm. So when we're talking about glycolysis, that was all happening in the cytoplasm, or the cytosol, basically the general inside of our cells. But then a lot of our stuff is going to happen in our mitochondria. And part of this is that whole like compartmentalization thing I was talking about, where basically we can control the concentrations of things to have different concentrations in different parts of the cell so that we can regulate, we can have conditions that are favorable for one thing inside of the mitochondria, say, but conditions that are favorable for something else outside of the mitochondria. The mitochondria, the reason why we're able to kind of like have different concentrations in there is because we've got membranes that are going to be selective. And so these membranes aren't going to let through everything. Most things can't freely go through. And even those things that can go through, they have to go through special transporters, all those different types of transport that we talked about. The mitochondria, it has two membranes, and the inner one especially is going to be very, very picky. And it's going to let through pyruvate, but it won't let out, and it'll change pyruvate into oxaloacetate, but it won't actually change, it won't let oxaloacetate through. So we have to go this convoluted route where basically we're going to take pyruvate into the mitochondria. Pyruvate carboxylase, that first enzyme that was involved, is going to then turn that into oxaloacetate. That oxaloacetate is then going to be converted into malate, which is then going to be taken out of the mitochondria where it can be converted back into oxaloacetate. So this whole complicated route to then have oxaloacetate outside, which now we can basically phosphorylate through PEP carboxykinase, and we can get our phosphoenolpyruvate. So long pathway, but we get there. So once we're there, we've got our phosphoenolpyruvate. Now we can basically, all these steps were easily reversible. So we can go back all the way up to here, but now we get to that fructose 1,6-bisphosphate, and we need to get to fructose 6-phosphate. So basically all we need to do is we need to chop off the phosphate group. But we're not going to use the same enzyme as we used before. We're not going to use that phosphofructokinase. We're not going to try to take that phosphate group off of the 6th position and stick it, or of the 1 position and stick it onto ADP. Instead, we're basically just going to cut that phosphate group off. So we're not going to get an ATP out of this. Instead, we're just going to cut that phosphate group off. And we're going to need a different enzyme to do this. We're going to need this fructose 1,6-bisphosphatase. We'll talk more about how this is going to be highly regulated, because remember, that was that committed step of glycolysis, and we can regulate this step. We can regulate whether glycolysis goes forward or whether we take things in the reverse direction. And then finally, once we get back past that step, when we have this fructose 6-phosphate, we can easily interchange that to glucose 6-phosphate. That was just one of our isomerization reactions. Very easily reversible, but now we have this glucose 6-phosphate and it's trapped inside the cells. Now our gluconeogenesis is happening in the liver, and the liver's job is kind of like, well, one of its jobs is to supply glucose to the rest of your body. And if we said that the glucose, the reason why we phosphorylated the glucose was to trap it inside of our cells, and now we're saying that basically it's phosphorylated, it's not getting out of the liver until the liver dephosphorylates it. And again, this case, we're just going to use a phosphatase, which is a hydrolase that's going to basically use water to cleave off the phosphate group to give us glucose, which can then get taken out of the liver. It can get into the blood, and other tissues can use it. So let's talk about how we can basically regulate these things. So remember, when would we want to do glycolysis? If there's lots of glucose, but not enough energy. And so how do we know if there's not enough energy? It's easy to tell if we have a lot of glucose, but how about energy? Well, basically, we would have low concentrations of ATP and high concentrations of ADP and AMP. So remember that AMP was going to be kind of like a more sensitive indicator of energy levels than ADP, because we had that we could take 2 ADPs and change them into ATP and AMP, and therefore we're going to kind of keep the AMP levels. The ATP to AMP ratio is going to be a better indicator, but we'll see that both ADP and AMP are going to be allosteric regulators. So they're going to be able to bind to enzymes and alter their activity. Another sign of not enough energy would be if we had low NAD plus and high NADH. Alternatively, if we had enough energy but not much glucose, we would have the opposite conditions, and we would want to make glucose through gluconeogenesis. You could also then like break down glycogen, but we'll talk about that later, but not right now. Right now, let's just focus on this glycolysis and gluconeogenesis. And so, before we can talk about which direction things are going to go once they're inside the cell, once glucose is inside the cell, we need to talk about how glucose is actually going to get inside the cell. Glucose transporters are going to be regulated through hormonal signaling. In response to insulin, which is basically a signal that's saying like, oh yeah, we just had a big meal or something, we've got a lot of sugar, we want the cells to let sugar in. Well, one of the ways that they can respond and let sugar in is by basically taking these pre-made glucose transporters in that we're kind of just like hanging out in these internal membrane vesicles and then transport them onto the surface. So in this way, we're able to control the import of glucose into the cells to begin with. And then once they're in the cells, that's where we decide, okay, do we want to trap them? Do we want to have this do we want to trap this glucose inside the cells so that we can use it for things? That trapping is done by this hexokinase. So we actually have 4 different versions, or what we call isozymes, so versions of this enzyme. Having these different versions is helpful because we can regulate them differently and they can have different activities. So hexokinases 1, 2, and 3 are all inhibited by their product, glucose 6-phosphate. So remember, this is one of the ideas we talked about before, where if you were like inhibited by your product, so you have like negative feedback, so that you don't keep making product if it's kind of like building up. If it's something that you don't need, then don't. And so basically these enzymes are going to be controlled in that way. Now we have a 4th hexokinase. This is also called glucokinase. Now this one's specific to our liver, and it's not inhibited by glucose 6-phosphate. And so it's going to basically, it would keep making glucose 6-phosphate. It's not going to be inhibited by that. One of the other things about it is it has different kinetic properties that make it especially good for being maintaining glucose homeostasis. So it has a Km that's higher than the usual glucose concentration, which is going to allow it to be sensitive to the glucose concentrations. And it's going to make it so that it's not going to hog all the glucose. So most of our cells have a hexokinase with a lower Km. So remember, the Km is the concentration at which half of it, at which it's at half maximal velocity. Our hexokinase 1, it's got this much lower Km, which means it's going to basically at lower concentrations, it's going to be like saturated. It's going to be fully active. However, with this hexokinase 4, remember that's also called glucokinase, here it has a higher Km, so it has a lower affinity basically. And so we need at the same concentration of glucose, it would be phosphorylating a lot less of it. This is going to make it so that it's kind of going to let through, it's not going to hog all of the glucose, it's going to let some out. Because remember, things kind of like go through our liver. And so and the liver is great for like detoxing and stuff because you can have all this stuff go through it, but we don't want it stealing all the glucose as it goes through. And so that's why basically we want it to be able to release some of this glucose. And so by having this higher Km, it's able to let the glucose through, and it's able to kind of be responsive to glucose levels. If you have this concentration of very low Km, well, basically, then you would always be at levels at which you were saturated, and you wouldn't be able to change activity in response to differences in the levels. Whereas with hexokinase 4, it's kind of in this range where you can then respond to differences in the levels. Our next step that we need to regulate is this big one, that fructose 6-phosphate to fructose 1,6-bisphosphate. So remember, in glycolysis, that's carried out by phosphofructokinase 1. And then to go the reverse way, we're going to use fructose 1,6-bisphosphatase 1. And these are going to be highly regulated and reciprocally regulated. So why are they so crazy highly regulated? Because, as I mentioned, this was the committed step of glycolysis. So basically, a lot of things glucose can go a lot of different directions once you trap it inside of the cells. One of the main other ways it could go, other than glycolysis, is basically is called the pentose phosphate pathway. This is going to be used to make NADPH, DNA, RNA, fatty acids, steroids, etc. So there's all these sorts of other things we can do with glucose rather than just break it down for energy. And so, but with either of those, we're going to need to trap the glucose inside of the cells, which is where the hexokinase is going to come in to play. So if we want to use glucose for any of those things, we're going to want to phosphorylate it. So that wouldn't be the main commit. That's not a committed step to glycolysis. Instead, after we do this isomerase step, the next step is the committed step, where we're basically actually saying, okay, now let's take this fructose 6-phosphate and change it to fructose 1,6-bisphosphate. In order to kind of regulate what direction things will go, one of the main regulators of this is going to be NADPH. We'll get back to the pentose phosphate pathway at the end, because that G6PD, it turns out to be medically important, and I'll tell you about that story at the end. So spoiler alert, stay tuned. But first, let's get back to how this is regulated regulated. And so one of the regulatory things is going to be NADPH. Because it turns out that one of the things, remember, of the pentose phosphate pathway is it's actually making NADPH. And so if you make a lot of NADPH, well, now we get this negative feedback regulation, which says, okay, we've done enough of this pentose phosphate stuff. But even once we make that decision, we still need to decide, okay, well, are we going to go towards glycolysis? Are we going to go towards gluconeogenesis? Do we want to upregulate PFK1, or do we want to upregulate FBPase1? And so PFK1 would basically take us towards glycolysis, and FBPase1 would take us towards gluconeogenesis. And these are going to be allosterically regulated by citrate, ATP, AMP, and ADP. So citrate, that's going to be basically one of the components of the citric acid cycle. So basically, if we were to go through glycolysis, downstream of this, we would have citrates. We'd also get that from fatty acid breakdown and stuff. So basically, if we're making enough energy, this is going to feed back and say, okay, PFK1, stop sending stuff down glycolysis. We've got plenty. But what if we don't have enough energy? We would still want to basically keep doing glycolysis. And so if we basically don't have a lot of ATP, if we have instead a lot of ADP or AMP, these are then going to go and stimulate PFK1 to take us towards glycolysis. And we could also say, okay, well, if we have a lot of AMP, basically we don't have a lot of energy, and so we're not going to want to go, and we're not going to go and make glucose instead of breaking it down. So let's inhibit FBPase1. So the same molecule, AMP, can regulate both of these. Another regulator of PFK1 as well as FBPase1 is going to be this molecule called fructose 2,6-bisphosphate. It's another one of those allosteric regulators, and it sounds like it's something that's just like from your glycolysis pathway. Well, there we had fructose 1,6-bisphosphate, and here we have fructose 2,6-bisphosphate. So when you see bis, basically this is saying 2, it's got these 2 phosphate groups. If you see diphosphates, like ADP, adenosine diphosphate, that would be the phosphate groups attached to one another in a row. But when we see bisphosphate, they're in 2 different places. And in glycolysis, we get this fructose 1,6-bisphosphate, and here we're talking about fructose 2,6-bisphosphate. Now, both of these are going to be made from fructose 6-phosphate, which was a product of the glycolysis. So this is kind of like a sidetrack from glycolysis that's going to give us this molecule, F26BP. Now, F26BP is able to allosterically regulate both PFK1 and FBPase1, but in opposite directions. So when it binds to PFK1, it increases its affinity for fructose 6-phosphate. And when it binds to FBPase1, it reduces its affinity for its substrate. PFK1 activity in the presence of F26BP is going to be higher, but the FBPase1 activity in the presence of F26BP is going to be lower. And so, well, how do we regulate the making of F26BP? So we said if we have F26BP, we're going to stimulate PFK1, and this is going and inhibit FBPase1. So this way we don't have this like fetal cycle. We're regulating them at both at the same time, and we're doing so in a way that's going to favor glycolysis. If we don't have F26BP, well, now basically things are going to be the opposite. We're going to be favoring gluconeogenesis. And so we can control the levels of F26BP accordingly. And this is done through hormonal control. So going back to our idea of like insulin, and insulin's kind of opposite, glucagon. So insulin is going to be released by our pancreas when we have a lot of glucose. Says let the glucose in. And then we have our glucagon, which says we don't have enough insulin, we don't have enough sugar. And so, hey, liver, stop eating the sugar and give us some. And start making it. So basically, we want gluconeogenesis in response to glucagon. If we want gluconeogenesis, well, basically what we want to do is we want to have low levels of F26BP. Whereas if we have insulin, lots of insulin, we're going to want high levels of F26BP. And so it turns out that basically what can happen is that the making of this F26BP is done by this really cool enzyme that's actually this two parts, or it's bifunctional. So it's got a phosphatase domain and a kinase domain in the same protein. It's really cool. So basically it has this kinase domain that's going to take fructose 6-phosphate, phosphorylate it to give you fructose 2,6-bisphosphate. And then it has a phosphatase domain, which is going to take fructose 2,6-bisphosphate and dephosphorylate it to give you fructose 6-phosphate. What determines whether it's acting as a kinase or whether it's acting as a phosphatase is its own phosphorylation state. When it's phosphorylated, basically, you're going to activate the phosphatase domain. This is going to then decrease the levels of F26BP, and this is going to inhibit glycolysis and stimulate gluconeogenesis. Alternatively, if it's not phosphorylated, well, now you're going to have the kinase domain active. You're going to make F26BP, which is going to stimulate glycolysis and inhibit gluconeogenesis. So if we phosphorylate this enzyme, we are basically going to be inhibiting glycolysis and stimulating gluconeogenesis. If we dephosphorylate this enzyme, we're going to stimulate glycolysis and inhibit gluconeogenesis. And so now we need to go back and think about when would we want to do what. Well, in response to glucagon, our signal that there's low blood sugar, we're going to want the liver to basically increase the amount of gluconeogenesis that's going on and stop doing glycolysis. So we would want there to be phosphorylation of this enzyme. Glucagon is going to act through a G-coupled protein-coupled receptor to make cyclic AMP. That cyclic AMP is going to activate the cyclic AMP-dependent protein kinase, which is then going to phosphorylate this protein, inactivate its kinase domain, activate its phosphatase domain, and basically then inhibit glycolysis and stimulate gluconeogenesis. To remove the phosphate, well, here it's going to respond to insulin, so basically the opposite of glucagon. The insulin is going to activate phosphoprotein phosphatase, which is going to remove this phosphate group to stimulate glycolysis and inhibit gluconeogenesis. So long, complicated, all this sorts of regulation, but is a great example of reciprocal regulation being used so that you don't have this like fetal cycle, making and breaking glucose at the same time. And finally, let's talk about this last step. In glycolysis, we take phosphoenol pyruvate and we take the phosphate group and off of it and put it onto ADP to give us ATP. This is carried out by pyruvate kinase. In gluconeogenesis, this is the one that happens in those two steps. We have our pyruvate carboxylase step that's going to take us from pyruvate to oxaloacetate. And our PEP carboxykinase step is going to take us from oxaloacetate to phosphoenol pyruvate. So that glycolytic enzyme, pyruvate kinase, it's going to be allosterically inhibited by ATP, as well as in our liver, we'll see that it's actually going to be regulated by phosphorylation as well. So we have different versions of this pyruvate kinase. We have in our liver, we've got this L form, and then like in our muscles, we have this M form. Both of those are going to be regulated by ATP, but then also in our liver, we're going to get this phosphorylation regulation, which we'll get to in a second. Why does it make sense to be regulated by ATP? Well, basically, if you take pyruvate, you can go different directions. You can go and you can take it through the citric acid cycle, and you can generate energy. You can make ATP. Or you could go and you could take it to oxaloacetate. You can go to gluconeogenesis and make glucose. Now, if you have a lot of ATP, basically the way you're going to get ATP is if you were to take it through to the citric acid cycle and then to oxidative phosphorylation that way. And if you have a lot of ATP building up that way, well, maybe it's time to go a different way. And so the ATP is going to negatively regulate this pyruvate kinase. In addition to being regulated by ATP, it's also regulated by other downstream things. So it can be regulated by acetyl-CoA, by alanine, which can be made from pyruvate, by long-chain fatty acids, basically a lot of different signals that we have enough energy. And so let's go and let's do other things like make glucose. It also gets positively regulated though. And so one of the things that actually regulates it is F16BP. So we just talked about how F26BP could be used to regulate things. Well, F16BP, that was actually one of the products of glycolysis. So basically up here we have this F16BP being made, and it's going to be activating this kinase at the very end of the pathway. Why does this make sense? Well, remember that this was our committed step. And so if we're saying, okay, we're pumping a bunch of stuff into this pathway, get ready for it. And so it's going to say, okay, get ready, pyruvate kinase, get active, because we've got a lot of stuff coming your way. And so that's why you get this kind of positive feedback from earlier on in the pathway, as well as the negative feedback from later on in the pathway. And then in our liver, things are going to be a little more complicated. We still have that same regulation, but now we're going to have an extra layer. Because in our liver, remember here, we can actually go and we can do gluconeogenesis. And so in our liver, basically, it's going to respond to glucagon. So it's going to respond to that hormone that says that we have low blood sugar. And our other cells, they're not going to have the glucagon receptors and stuff, but our liver is. And that glucagon receptor is going to act as one of those GPCRs. It's going to go through and, like we talked about before, give us the pathway where basically we can make cyclic AMP. That's going to activate our PKA, our protein kinase A. That's going to phosphorylate pyruvate kinase, the liver form. Now, when it's phosphorylated, it's inactivated. So we see a lot of instances in which phosphorylation activates something. In this case, it deactivates something. So it deactivates pyruvate kinase. If you're deactivating pyruvate kinase, well, here, this is going to prevent things from the pyruvate from going into glycolysis, and instead let it be used to go the other way and make glucose from it. And so that's how we can regulate the pyruvate kinase. How do we regulate the other things? So we have our pyruvate carboxylase and our PEP carboxy kinase. Basically, pyruvate carboxylase, so that one that was taking pyruvate to oxaloacetate, it turns out that it is going to be allosterically activated by acetyl-CoA. So remember, acetyl-CoA was going to be inhibiting the pyruvate kinase. And so it was inhibiting the upstream path of this from glycolysis. It's also actually going to be inhibiting the step from pyruvate to acetyl-CoA, so that things are kind of like, if you do get to pyruvate, don't go forward into glycolysis if you already have a bunch of acetyl-CoA. But if you have a bunch of acetyl-CoA, okay, now let's stimulate it going the other direction. Let's stimulate that pyruvate carboxylase to get us oxaloacetate. In terms of, though, we still have that next step where we have to go from oxaloacetate to our phosphoenolpyruvate. We need that PEP carboxy kinase. And it turns out that it's mostly going to be regulated transcriptionally. So our insulin, it didn't just activate that one kinase and stuff. It actually goes through pathways that are going to activate transcription factors. So it's going to activate proteins that are going to regulate the expression of genes. And it's going to regulate the expression of a bunch of different enzymes, including decreasing the expression of PEP carboxykinase. So it's basically insulin is then able to turn off the things that you need for gluconeogenesis and turn on things that you need for glycolysis. And then we have other hormones, other transcriptional regulators that can stimulate making things for gluconeogenesis. So there's all sorts of transcriptional regulation that goes on there. Finally, as I promised, I want to tell you that cool medical story about the pentose phosphate pathway. So basically, when we have the pentose phosphate pathway, that's when we want to make NADPH. We want to make DNA, RNA, fatty acids, steroids, all those good things that we can make from glucose instead of just like breaking it down. The cells and tissues that are going to use it, these include like rapidly dividing cells so that they can make RNA and DNA so that they can duplicate their DNA in order to divide and still have enough DNA for each cell. Tissues that are doing a bunch of fatty acid synthesis, that fatty acid synthesis is going to need that NADPH. So remember, we talked about how NADPH is going to be used typically for anabolism. So for making things, well, we're going to have to regenerate that. And one of the ways that we can regenerate that is through the pentose phosphate pathway. Another reason why you would want to use the pentose phosphate pathway is if you're making cholesterol and steroid hormones, which are also going to need that NADPH. And remember that NADPH was kind of going to be what was the relative concentrations of that were kind of helping control whether glucose 6-phosphate was entering glycolysis or the pentose phosphate pathway. And so if you already had a bunch of NADPH, then you don't need to go to the pentose phosphate pathway, at least for that sense. So that would inhibit this and stimulate glycolysis instead. But that's not all that you need the pentose phosphate pathway for. But the NADPH really is important. And one of the things that it's important for is actually regenerating glutathione. So glutathione is one of our body's main antioxidants that basically it's able to take the hit from reactive oxygen species to oxidize. So you go from these two glutathione molecules to oxidized glutathione, where you form this disulfide crosslink between them. In order to regenerate glutathione so it can be used as an antioxidant again, you need to reduce it. And it's reduced by NADPH. And this NADPH is made by this pentose phosphate pathway. And so what's the medical connection? Well, it turns out that this G6PD is this glucose 6-phosphate dehydrogenase. It's one of these enzymes in the pentose phosphate pathway. So it's this first enzyme in the pentose phosphate pathway, and it makes NADPH from NADP+. And it turns out that some people have a deficiency of this. So you might have heard those commercials like, ask your doctor if you have a G6PD deficiency. Well, the problem with having a G6PD deficiency is now you can't keep up with oxidative stress sometimes. And this is especially potentially an issue in the case where you're taking like anti-malarial drugs or other drugs that lead to an increase in reactive oxygen species. So that reactive oxygen species can kill the malaria parasite, but it can also overwhelm the red blood cells of patients with this deficiency, causing those cells to degrade and leaving the patients without enough red blood cells. So this is called like hemolytic anemia. There's also other chemicals that can trigger this, as well as like including ones that are found in fava beans. So people with this deficiency should avoid such drugs and triggers, but many people don't even know that they have the condition because it's not routinely screened for. But that is your G6PD connection, which again was that enzyme, the first enzyme in the pentose phosphate pathway, which was an alternative fate for glucose. So remember, metabolism is messy, but there is a logic to it. And if we focus on this logic, then we can understand all sorts of different metabolic quote unquote pathways, which are really more like this giant web. So focus on the logic of metabolic pathways, the chemical logic. Why do we shift things around? Why do we phosphorylate here? Do we want to make a better leaving group? Do we want to set things up for a cleavage? What about the thermodynamic logic? How can we set up pathways so that we can use energetically favorable reactions to drive unfavorable ones? How can we couple things together? How can we control the levels of the concentrations of things, keep things in a steady state at which we're able to continuously have a drive, but have steady levels of things? How can we maintain some reactions near equilibrium so we can easily reverse them, but other ones far from equilibrium so we can use them to control directionality based on controlling the activity of the enzymes? We can control the activity of the enzymes on different time scales, controlling when these enzymes are actually made on a longer time scale, or quickly regulating them based on like phosphorylation, sometimes in response to a signaling cascade, based on allosteric regulation, sometimes by the things that upstream in the pathway to so that you get ready down the line, sometimes from a negative feedback from things further down in the pathway, so that if you're making enough of something, you don't keep making it. And we could also kind of free up enzymes from sequestration, take them, say, out of the nucleus and into the cytoplasm. All sorts of different examples of how we can regulate these pathways by regulating the activity of the enzymes, as well as the concentrations and all that good stuff. So metabolism is complex, it's intertwined. We've looked at these pathways in kind of like isolation, but really it's all connected.